I really like the analogy of Velcro. Where would we be without Velcro? And it was a serendipitous finding by some person who realized that burrs stuck to certain materials. And so I'm interested in puzzles. I'm interested in how cells normally function.
Mm-hmm.
You have to understand how cells normally function in order to understand disease, I think. Otherwise, there's just a correlation between, well, this happens in the cell and there is this disease, but it's a black box in between the two pieces of information.
I'm John Foxe, Director of the Monte Institute for Neuroscience at the University of Rochester, and I'm delighted to welcome you to another episode of Neuroscience Perspectives. I'm absolutely honoured to be joined today by a leading mechanistic biochemist whose discovery of a cellular quality control mechanism has literally transformed our understanding of a wide range of disorders and created new avenues for development of personalised treatments. Dr. Lynne Maquat is the Director of the Centre for RNA Biology at the University of Rochester and the J. Lowell Orbison Endowed Chair and Professor of Biochemistry and Biophysics. Lynne discovered nonsense-mediated mRNA decay, don't worry, we'll get into it, or NMD for short, one of the most prominent surveillance systems in the body to protect against mistakes in gene expression that can lead to diseases like cystic fibrosis, Fragile X, and different types of cancer. Lynne is the recipient of numerous awards. We'd be here for the entire episode if I was to list them all, but these include the prestigious Canada Gairdner International Award, the Albany Prize, the Wolf Prize, the Dr. Paul Janssen Award for Biomedical Research, and the Gruber Genetics Prize, and that's just a small little sliver of them. She is dedicated to uplifting and mentoring the next generation of scientists, and she is the founding director of the Graduate Women in Science program at the University of Rochester, a group that cultivates mentor opportunities and personal and professional development for all. Lynne, I'm absolutely delighted to have you here on Neuroscience Perspectives. It's a great honor, and you and I go back a bit, and
we do.
We're friends as well, and it's been marvelous for me to witness the just evolution of the work and it getting such great international recognition. Now, we hit folks in the opening there with some complex language.
Yeah.
And there's probably not a person on the planet at this point who hasn't heard the term mRNA or messenger RNA. But your work really goes to a next level and looks at this nonsense-mediated decay.
Right.
We're great at our jargon in this field. So you've got to help out the regular listener out there. What is nonsense-mediated decay of RNA and why should they care about it?
Okay.
All right.
mRNA, I think, has become more of a household term because of the vaccine for the COVID pandemic. And so what I've done for many decades is understand how cells make mRNA. It's a very complicated process. And as we have checks and balances in many aspects of our lives, cells do the same thing. NMD, I'll call it NMD for simplicity, is one of these checks and balances that the cell uses to make sure the mRNA is not going to make a truncated protein, which could be bad.
Yeah.
It could be toxic to cells.
Right.
And we discovered this by studying human diseases because a third of all inherited and acquired diseases have NMD targeting the disease-causing gene product.
Gotcha. Let's back up just a little bit. There's a lot. We have a wide variety of listeners here. So tell a little bit about what messenger RNA does in the first place and how that works, just so people can understand this business of these truncated proteins.
Okay. Messenger RNA, mRNA, encodes protein. Mm-hmm. And so the mRNA is made in the nucleus of the cell. That's where the genes are, through a complicated series of steps. And then it goes out into the cytoplasm where the protein synthesis machinery exists, and that's where it makes protein.
And we're built of these proteins. These are the essential building blocks of a human being, right?
That's correct.
And other things, but they're very important.
They're very important.
And so if this process goes wrong and the protein is truncated, shorter, or doubled up, or has some other conformational change, we're on the road to bad news.
It's like throwing a wrench into a supply chain where the whole chain will stop. And the way the cell addresses this is early on, before the protein can be made by the mRNA, by degrading the mRNA itself.
Right. So this is really a check on the system. Don't make that. That's bad news for this. And when that process of breaking down the mRNA doesn't work, we're on our way to some important diseases that we know about.
There can be diseases that are inherited in a dominant way. So you just need one defective allele, either from the mom or the father, to cause disease. And we know the rules for which mRNAs will be causing a recessively inherited disease versus a dominantly inherited disease.
Fantastic, yeah. And so this leads a structural biochemist right over into the world of neuroscience with intellectual developmental disabilities, and particularly Fragile X syndrome. And you've done quite a lot of work there. Do you want to speak a little bit about that?
Sure. If you had asked me, let's see, maybe 6 years ago, if I were going to be working in neurobiology and on Fragile X syndrome, I would have looked at you like you were crazy.
Right.
However,
which I think you have a few times, but we won't get into that.
But for different reasons.
That's true.
So we were looking for new proteins that functioned in NMD. We can learn a lot about how cells do what they do by looking for protein-protein interactions. What we did is we looked for proteins that interact with the key NMD protein, and we found the protein missing in Fragile X syndrome. Well, what does that mean? We thought a lot about it. Both proteins are RNA binding proteins. So remember, I'm an RNA biologist.
Yeah.
I'm all about RNA. This was very intriguing to us, and we were very interested in Fragile X syndrome because there's no cure.
That's right.
So we thought, getting at mechanism in a way people hadn't appreciated would be helpful.
Right.
We worked, because I'm not a neuro person, we worked with Chris Pichel, who's also at the medical center. He's a stem cell biologist. He's a neurobiologist. He's a mouse developmental biologist. And he taught Tatsuaki Kurosaki, who was a research assistant professor in my lab and now runs his own lab at SUNY Buffalo Medical School, how to work with patient cells, skin cells, to de-differentiate them to cells that could be differentiated into neurons. And we could understand from an RNA perspective and from an NMD perspective what goes wrong in Fragile X syndrome.
I mean, it's a fantastic tour, right, from this fundamental understanding of molecular biology, of nonsense-mediated decay, all the way over to taking a biopsy from a patient with Fragile X from their skin, and then turning those into neural progenitor cells, stem cells.
That's right.
And growing neurons and being able to actually go after the disease process in the cultured neurons. It's a tour de force.
It's amazing. And then fortuitously, it was as if the stars aligned. I was mentoring an MD PhD. So this is a woman who's a researcher physically in my lab, and also an otolaryngologist who had interest in the Fragile X mouse. Mice can serve as wonderful disease models. And she brought the Fragile X mouse model to our lab, and we could understand when problems in Fragile X syndrome start. And they start in utero, but they persist postnatally after the mice are born. And we are now searching for small molecule inhibitors of NMD that could be drugs. So we have inhibitors, but they're not user-friendly in the clinic, to see if we can use this window of time during which NMD is hyperactivated, it's too active, to dampen that hyperactivity and possibly help patients.
It's extraordinary. Just again, for the listener out there who may vaguely have heard of Fragile X syndrome, say a little bit about it, right? I mean, it's a leading cause of autism, one of the most penetrant versions of it.
That's right. So autism is a very complicated disease. And in the case of Fragile X syndrome, there's a single gene cause in most cases. So therefore, there's a target. It's easier to study the process. And this is a disease that is diagnosed in our country at around 3 to 6 months of age. And the kids just don't, it's very diffuse in how it manifests in terms of severity, but generally the kids don't thrive in society.
Yeah, amazing. I mean, and again, going to this multi-pronged approach from the original work that's pure discovery, pure biology, no disease necessarily in the crosshairs, all the way to a mouse model and small molecule inhibitors that might actually ultimately end up in the clinic and help us with a leading cause of autism. I think sometimes our general listenership don't appreciate the span of work that needs to be done, and the fact that some of these discoveries, they date back decades in terms of where the original work was done.
Right.
Can you speak to that, the need for basic research that doesn't necessarily at this moment in time have a disease in the crosshairs?
Sure. I really like the analogy of Velcro. Where would we be without Velcro? And it was a serendipitous finding by some person who realized that burrs stuck to certain materials. And so I'm interested in puzzles. I'm interested in how cells normally function. You have to understand how cells normally function in order to understand disease, I think. Otherwise, there's just a correlation between, well, this happens in the cell and there is this disease, but it's a black box in between the two pieces of information. And so I began by understanding how cells normally function with a focus on RNA, and was interested in the thalassemias. I loved taking my strong background in biochemistry, and finally, after undergraduate lab work, graduate lab work, and now doing postdoctoral lab work, applying it to a disease, not having any understanding of where it was going to lead us.
Right.
And I have to say that I feel very fortunate to have been at the right time, at the right place when technology was available to figure out what was happening. And that was the first definition of NMD.
Yeah, and I mean, in your previous description, from the stem cells to the tool chest that we have available today, is just extraordinary, right? But again, coming from basic discovery. Basic
discovery when technology was... Science is very labor intensive, but technology back then, it took us months to do what we can do now in days.
Yeah, exactly. Yeah, I love your analogy of the burrs and Velcro. I always say, I have it. My own story is, you wouldn't bring your car to be fixed to somebody who didn't know how an engine worked in the first place, and you'd really need to let our field be busy about just figuring out how the engine that is your brain works in the first place. Because until then, we really won't be in a position to fix things the way we'd like to.
That's right. And when you think about yeast, baker's yeast, you use it to make beer and other things. The studies of yeast in the lab helped us understand the cell cycle and what is cancer, but it's an abnormal cell cycle. So the basics of how organisms, they can be model organisms. Eventually we can translate that to humans because there are many similarities, are very helpful in laying the groundwork.
Right. Maybe another example, of course, is, mRNA vaccines. COVID arises, and within a matter of months, not too much more than a year, we have a vaccine. And I think, again, there are many people out there, and there were narratives in the body politic about it as if the vaccine had been developed overnight. But this was on the back of decades of work.
Many people. So people had, my lab, way back, decades ago, were injecting or introducing mRNAs into cells. But one of the keys to the vaccine, and again, this was realized many years ago, was the RNA needed to be modified so that it was stable, translatable, and wasn't recognized as a foreign agent, so our immune reactivity would be to the mRNA and not the protein that it encoded.
Right.
And very few people talk about delivery systems. It's been the mRNA that's gotten the limelight. But don't forget, you can have the mRNA, and the way to introduce it into cells, though, becomes a problem. So there were many people working for many, many years on lipid delivery mechanisms. This was not an overnight discovery. And fortunately, for the COVID pandemic, scientists were in a position to be able to address it.
Yeah, exactly. Decades of work, the right technology, poised to go. We get hit with a pandemic, and it really is a matter of months. It's a shame to hear such suspicion. You must have thoughts about this, with the current political zeitgeist, the vaccine hesitancy. What do we have to do actually as a field to be better about communicating how extraordinarily useful these things are and safe?
And safe. Well, it's certainly better to get a COVID vaccine than to get what is called long COVID. That's a very bad disease. Mm-hmm. And I think scientists throughout history can be considered, under certain circumstances, as outliers. Remember, people didn't want to believe that the Earth wasn't flat. People don't want to believe that smoking causes cancer. These are messages the scientists are telling people. Yeah. And today, many of us don't want to hear that we probably shouldn't be drinking so much alcohol.
Yes, unfortunately.
But, those findings are based on scientific discoveries and careful science. I think the problem with the COVID vaccine and the fear came from the idea that it's new. It is new, but also that it came out of nowhere, and that's not true.
Yeah, right. And lots of conspiracy theories and so on.
Well, and the problem with social media, I believe, is once you tap into a certain vein of social media, you will be getting information that backs that view rather than seeking broadly. And I have to say, that's what scientists do. We can have a hypothesis, but the goal is not to prove the hypothesis. The goal is to determine if the hypothesis is correct or not, very importantly. And we ask the question behind the hypothesis from more than one experimental way to be sure that we're not just getting an answer that pertains to a particular circumstance.
Right, going down some cul-de-sac that's self-reifying.
That's correct. Yeah.
On the messaging, so I'm glad you brought up social media and this kind of iterative minimization thing that happens. People end up down their own little alleyway. What can we do? I mean, are we equipped in the sciences to fight back against this kind of misinformation? Should we all be getting media training? Do you think about that kind of stuff?
Yes, and that's part of what one of the programs that I started here and still lead does. So we have an NPR reporter, science reporter, who's going to talk to us next week, Pen Huang, about how to speak to the lay public.
Yeah.
And I encourage students, we've had little contests for students and postdocs. How do you speak to your family of non-scientists about what you do? We have our own language. Scientists have their own language. We speak a lot of jargon, and it's very important for the process. But when we then want to go broadly and reach out to people who are not scientists, it's important for us to be able to communicate clearly and better.
Jargon is a big problem, and I think one of the things that I'm profoundly aware of is that once folks know that you're a scientist, they worry that they're going to be talked down to, and then we start in with the jargon, and it sounds like we're talking down to people. Yeah. And for us, it's like having a language. It's like Spanish or English or whatever our natural language is. We've lost the part where that language was developed over the course of decades, and we think it's amenable, it's open to folks, but it's not. And trying to get back to where people are. We do have complex things that we need to explain. But I mean, I really think in your case, the work is very complicated because you're really in the nuts and bolts of molecular biology. But you do a great job of pulling back out, and doing a 30,000 foot look at it. And that's very important. So you're a molecular biologist. We want to know about you, Lynne. Like, when did it start? What was the young Lynne Maquat like? And when did you get biology in your crosshairs? Did you grew up in the United States?
I grew up in the country in Connecticut, and my mom was an RN, and my father went to night school after World War II and was an engineer. And for years, I thought he drove a train. I have to admit, I was young, but I thought he drove a train. In high school, I was very shy and quiet. I got the Spanish award and the English award and didn't particularly like science. I went to the University of Connecticut because we could afford it.
Yeah.
And I got into a cell biology lab course. Very different than the science in high school. We were studying, believe it or not, how mRNAs make protein by doing experiments in the lab. And it was a mechanism. And I thought, whoa, this was so cool.
Yeah, amazing,
So,
yeah.
I asked that professor if I could work in a lab, not knowing I could. I don't know what drove me.
Yeah.
And he pushed me to define what I meant by a lab. And so I squeaked out his lab, and he said, sure.
Very good.
Now, I didn't get mentoring as much as I could have used in that lab,
Sure.
but I was always afraid I was not well, it's hard in the beginning because most everything is new and it's hard to understand. I found the lab very intimidating, but I still loved it. I liked the challenge, and I liked that you had to know a lot to do anything. So the literature, reading the literature, plus the technology. What do you do with your hands? You have to do the experiment then.
Yeah.
Very complicated, but very cool.
Yeah.
It did not breed a feeling of confidence, but for some reason, I had the stick-to-itness that has served me well throughout my career.
Right. A singular focus and a roll up the sleeves and get stuck in kind of approach.
Yes, even though I was unsure,
Sure.
I thought this was the best approach. And so I remember that semester I had to write a essay for the cell biology lecture. And I remember going to the Yale Library and looking up papers on what I thought would be an interesting topic. And I remember saying to myself, this is either very good or very bad. And I couldn't tell because I was so young.
Yeah.
And it turned out it was very good.
Wow.
But
so right out of the gate, undergraduate lab, and you hit on some really good ideas.
I did, but I just want to emphasize the fear factor.
Sure.
I
Everybody's
lacked
intimidated.
confidence.
Yeah.
Oh, yeah.
Yeah. Yeah. I always say that when I'm talking to youngsters, imposter syndrome is never going to go away.
Never.
You'll have it to the very end. Doesn't matter how much success, how many medals or prizes you win. You always feel like, oh God, do I really belong here?
I feel better because the years have built on one another.
Yeah.
And where I was getting pushback from new ideas, they have proven to be okay. They've proven to be correct.
Right.
And that helped me a lot. Plus, I had people in my lab, so I do not do science by myself. I train graduate students and postdocs and technicians and undergraduates who believed in what we were doing. And then when I have colleagues who are in the trenches with me, even though they're in training, these are intelligent people who can provide their own perspective. So we talk a lot about how to design experiments and what does this result mean, and we refer to the literature. So we together work on developing a base of information that we can believe in.
Going back actually early in our little interview here, you were talking about coming at things from multiple lines of evidence, the scientific approach, the paradoxical approaches. And you're talking about having good ideas, but were there things that failed, and then how did you manage?
A lot of times things failed because the technology wasn't there.
Gotcha, right.
And another important aspect about research science is collaboration. And so I had data indicating through very tedious experiments where NMD was occurring in the cell. Careful cell fractionation studies, for example, and experiments that involve turning on a gene and shutting it off and then watching what happened to the mRNA as a function of time.
Right.
But a colleague of mine at Einstein, your colleague at Einstein, Rob Singer, and I were very close friends, and I bugged him for many years. Rob, can we do this experiment? He is an amazing biophysicist and microscopist. And eventually we were able to watch where NMD occurred in the cell. It was amazing. And thank goodness we were correct. And even my buddy, Rob Singer, didn't quite believe I was correct. He only told me that afterwards because he knew better. Yeah, but it was wonderful.
Well, and one of the great characters in all of science, Bob Singer. He's
a great guy.
Such a great guy.
Yeah.
Talk to us about the creative process. How much of science is doing the next obvious thing, and how much of it involves contemplation and trying to think outside the box? How do you approach that?
Well, I can quote my husband. Lynn, you look normal, but you certainly aren't. And I think he means the way I think.
Yeah.
And I think there is something to be said for not being entrenched in knowledge that pre-exists in a field that one is entering. And so I have entered new fields many times because of what we've discovered. And it served me well to ask questions that other people thought were answered, but in fact had not been experimentally addressed.
Yeah.
And the same goes with Fragile X syndrome.
So question all suppositions. Don't take anything as a given.
It was a supposition, but to me it was an obvious question that because we had tools we had built in my lab, no one was able to ask.
Gotcha.
And no one asked. And so I suggested, since we knew how to do it, to ask this question just to check. And we also had some indication that we could be right with this new idea.
Yeah.
I don't know how to teach creativity. I've been asked to, and I think it requires knowing what you know and knowing what you don't know.
Yeah. I have some insight into your work ethic, and I know how much time you spend in the lab and all the rest of it. But how much of it is contemplation? Like, what is there a mix in life, and then how do you approach that balance in life? And do you ever yourself get to leave the science behind, or is it just all consuming for you?
When I'm walking the dog at Corbett's Glen, my mind is free, and she brings me great joy, and we talk to everyone, and I have peripheral vision. When I'm in the shower, so I tell everyone in my lab when they join the lab, you'll know you're there when you're in the shower and you realize, you realize something. So I think it's always in the back of my mind. Sometimes I'll wake up, or I have trouble getting to sleep. And part of it probably is because the day seeps in, I realize what happened and what didn't happen, and then ideas come.
Gotcha.
When I take a vacation, I still need access to internet so my day-to-day operations don't fall short.
Gotcha. Yeah.
But again, there's an opportunity for more creative thought. Being on an airplane, a different environment, put me in one of the lounges at an airport. So I'm there for 8 hours. And if my husband is with me, he's tortured, and I'm like working away. Yeah. So I think different environments help.
Yeah, just shaking
open
it
the
up
mind.
A bit.
Yeah.
And travel? Does travel help?
Travel helps.
Yeah.
I've never been told something by someone that I haven't thought of already. They may tell me it, and I'll think, oh my gosh, why didn't I think of that? And then I'll realize I had. But still, it's an important exercise, talking to other people and being in different environments. And it's great to talk to people. Like, where would I be without talking to neurobiologists in the Fragile X project? I wouldn't be anywhere close to where we are now.
Let's talk a little bit about mentorship. And I want to bring up one other thing. It's always interesting in a world, particularly like, in molecular biology, which would have been quite male-dominated, I'm assuming, when you came into the field. Navigating that world as a woman, how has that been? And how has that then led, I know, founding this women's science movement and so on, how's that led to changes in the way you approach mentorship?
Yeah, it's greatly influenced the way I've mentored. Well, get the violins out. There were no female faculty members when I was a graduate student in biochemistry in Wisconsin. Great department.
Yeah.
But
not a single one.
No, when I was a graduate student. And I did not start out with a tenure track position. I probably could have if I asked for it, but I was afraid to rock the boat.
Gotcha.
And I think my main goal was for people to take me, other scientists and administrators to take me seriously.
Yeah.
And that's something that has to be demanded, but in a kind and compassionate way.
Right.
It's not helpful to go into an administrator's office and rant and rave. And I think always doing quality science, always staying funded. There's nothing that people can say about your integrity and ability to be a scientist when you have that.
Yeah, yeah.
And so, because it was a bit of a rocky road for me for a long time, I have established Graduate Women in Science here, which was initiated to address the leaky pipeline that still typifies women.
Yes.
Fewer women compared to men who have a PhD degree go on and use that degree in a career. There are many things you can do with a PhD in science besides being an academic researcher like myself. And so we bring in people from all different venues of what they do for their job. And I think it helps graduate students. It's open to all genders. Men need mentoring too, to see how people navigated life. Life is not going to happen the way you think it will, and that's probably a good thing.
Right.
And how people failed, both in personal and professional aspects of their lives, and resurrected from the failure.
Gotcha. Yeah. Maybe this is a slightly unfair question. You're a strong personality, Lynne. I mean, you're a great personality, and in those early days, could you be Lynne? Could you be you, or was there an accommodation to the environment? Did you have to?
I was shy and quiet.
Right.
Very shy and quiet. I was afraid to rock the boat.
Yeah, yeah.
And I was so shy that I would be so nervous when I gave a talk.
Right.
But I would be mad at myself if I didn't give the talk.
Right.
So it was a fine edge of pushing myself, which is how I've come to tell my mentees that it's okay to work outside of your comfort zone as long as you believe in what you're doing. And I realized many people tested me, and I would have to push back in a kind and caring way.
Right.
So, for example, I was organizing 2 meetings. I advise against it strongly and was writing 2 grants. And I needed help in the office. And so someone in an administrative position told me that I probably shouldn't be doing all of that because didn't want to give me help. And so my response was, why don't you think about what you just said to me and report back? And so that was a Friday. And on Monday, he apologized and gave me what I needed because it's all a win-win for everyone.
Yeah.
It's good for Rochester. It's good for everyone who attends the meetings. It's good to be funded.
Yeah.
And so, I realized that I need to push back in a caring and compassionate way because the motivation behind doing all of this is good.
Right. Well, you've had an extraordinary career to this point, and there's no slowing you down. There's wind in your sails. The work goes on. I wish you all the best. I love having a front row seat to watch the Lynne Maquat show. It's wonderful. Thanks so much for coming in and joining us on Neuroscience Perspectives. It's been a great honor.
It's been my pleasure, John. Thank you so much.